welcome to Technology Network's video podcast. Today, we are diving into the exciting world of scaling single-cell omics with automated liquid handling. Joining us are Giovanna and Paul from Scale Biosciences and SPT LabTech. Welcome to you both.
Hello.
Thanks for having us.
So, to get started, before we delve a bit deeper, I was wondering if you could each briefly introduce yourselves and tell us a little bit more about your roles. Paul, if you wanted to go first.
Yeah, so hi, I'm Paul Lomax. I'm head of genomics at SPT LabTech. And essentially that's responsible for all automation systems that we supply for genomics applications. And typically that's things like NGS library prep, PCR setup, and so on. And now, excitingly, the single cell RNA sequencing kit from Scale. So I've been in automation for quite a long time, goes all the way back into the 1990s. And even back then, I was working with customers who were involved in the human genome project. So back then, automating Sanger sequencing. And now in recent years, very much kind of focused on the growth of NGS and NGS based techniques, which has led to a lot of need for automation.
Fantastic. And Giovanna, what about yourself?
Yeah, so I am the CEO of Scale Bio, and I have spent my career in life science tools. So I started out at Illumina in development and operations, and then ended up running their NGS library prep portfolio. And so even in that portfolio, we were very keen on automating those workflows, like Paul had mentioned. And then I went over to 10x Genomics, where I was on the commercial side of launching their single cell portfolio, and ended up running commercial marketing, and then hopped over to Scale Bio, where I get to continue my passion for single cell omics, and driving new innovations in that market.
Fantastic. So, Giovanna, single cell research has become a bit of a hot topic in life sciences. Why would you say that these sort of studies are so important, and what kind of unique insights do they offer researchers?
Yeah, so single cell omics, single cell genomics has actually been around for quite a long time. It started back when people were interrogating one cell in one well, and they were realizing that there were significant differences in cell types per cell types, that could give us a better understanding of disease, could give us better understanding of just human biology in general. But the tools have evolved over time so we could interrogate more cells and more samples. And about 5 years ago, maybe 6 years ago, there is new technology innovations that really allowed the community to interrogate single cells at the level that needed to happen at that time. But we're really at this plateau moment right now with the current technologies, where we need to be pushing the limits in terms of the number of cells that can be interrogated, the number of samples that can be interrogated, but also the different omics that can be learned from a single cell to have a more complete picture of biology, to really drive understanding of disease and precision medicine. And so it is a really hot topic area right now, and it hasn't reached its max yet. So we're excited to be in this area.
Fantastic. And I guess perhaps there must be some challenges as well. What would you say are some of the key obstacles that are faced by labs in the field? And how would you also say, double prong kind of question here, how would you say that these challenges have driven the development of Scales technology too?
Now, there's a number of challenges, and we are in the research community, and so every researcher has very, individual needs for their experiments. And it's a platform that can be used across so many different areas of research too. And so the main challenges that you can tease out from all of these different experiments are people do want to run more samples or more conditions or more cells, because it just gives much more power to the studies, and they can be efficient with their dollars too, especially in the research community. There are only so many dollars to be spending to all of these cool new tools. And so the more information we can get out of these studies with a reduction in cost will only benefit science.
Absolutely. And I guess automation actually seems to be playing a bit of a big role in overcoming some of these challenges. So I guess a question to both of you is, could you elaborate on the advantages that automation brings to single cell sequencing library prep? And what specific challenges would you say that it solves? Perhaps we could go to Paul first.
Yeah, there's many elements to that. So Giovanna's talked about the increased sample numbers, increased throughputs, and automation obviously brings that. Some of these reagents are expensive, so the ability to pipette small volumes, dispense small volumes, can be challenging to do manually. And we've got systems that can do that more accurately through automation. And I think a really big one is consistency, from run to run, from user to user. Two users may not run a protocol the same way and get consistent results. So I think there's real advantages to that. And somebody might work better in the morning than say the afternoon. So there's even things like that come into play. And actually, some of these techniques have lots of pipetting steps in, and to do it manually, there's only so much you can do. You hit a limit as a human of how many samples you can manage. So it doesn't well scale, which is where we go with automation is generally trying to push more and more samples, higher throughputs. And people, scientists running these experiments didn't really study to stand at a bench all day pipetting. They want to be looking at the data, analyzing, and so on. So automation frees them from that and allows them to focus on the really important parts.
Nice. And Giovanna, what about you?
Paul covered a lot of the main advantages here. What I'll add is when we look at automation partners, we really want to ensure that we have partners that just don't focus on the ultra high throughput studies, that really have platforms that can be used by anyone, because maybe you do have a smaller project, but those pipetting steps can add in batch effects or are just tiring and there's not the consistency. And we just want to make it as easy to use as possible. So, like Paul said, those researchers can get to the data, they can find the biologically interesting information and spend their time there versus spending their time really at the bench.
Nice. So now let's talk about Scale's Firefly platform. Paul, could you explain the features and capabilities that make Firefly such an ideal solution for library prep in single cell sequencing?
Yeah, so we've combined a number of technologies into quite a small compact unit, and in a nutshell, they are pipetting, dispensing, shaking for mixing, incubation, and then kind of put that together with a very easy to use interface. So it's essentially doing what the person would do, walking from tool to tool, using different pipetting and dispensers and so on, incubators, all in one system that can actually just handle the workflow from end to end.
Nice. That's great to hear. And it sounds like a significant advancement. But Giovanna, could you walk us through the typical manual workflow for library prep compared to what Firefly offers? Where do we see the biggest gains in that productivity with automation?
Right. So in the workflows that Scale Bio provides, where we don't need any specialized instrumentation. So we're not forming droplets for single cells. We're processing the single cells in parallel in your standard 96 well and 384 well plates. And there's multiple steps within this workflow where you're pipetting either reagents or cells into a plate, you're extracting them out of a plate, you're putting them in a new plate. And so the manual workflow does have many pipetting steps, and pipetting steps within a 96 well plate, and in our current workflow in a 384 well plate where you're dispensing into. And that takes attention to detail to ensure that you're going to do it right. And many people pipette into and out of these types of plates, but again, we want to simplify it as much as possible. So since our workflow is plate based, And yes, it can be done manually. It is an ideal workflow to put onto a platform like Firefly because it absolutely simplifies and ensures the consistency and ensures the right well of reagent dispensing. And so we see, a huge difference in hands-on time when we use an automation platform like Firefly compared to someone doing something manually, and a lot
Fantastic.
less carpal tunnel in there too, from
I
my
can
buddy.
imagine.
Yeah.
So, I guess whilst automation is great, not every lab perhaps has that access to an in-house automation expert. So, Paul, how has this user friendliness been factored into the development of the Firefly workflow?
It's been absolutely central to the development of the product. It was one of the really key things when we first put the specification together and designing the system. We've that was really, really number one, both in terms of actually physically using the machine and, very importantly, programming the system. In the past, I've worked with customers with systems provided by previous companies that I've worked with, where we would run training courses, advanced user training courses, 3-4 days long, to train the expert user. And we really want to change things. We did not want to go there. We wanted it to be a couple of hours and somebody's able to do it, and that's what we're seeing with Firefly. It's really intuitive, it's very easy to use, and customers get up to speed with it really quickly. Um, because one, the expert may leave, the expert goes on holiday. Um, I've seen robots gathering dust in labs because the expert left. We did not want that, so we've really changed things with Firefly, and our customers are finding it really easy to use, and kind of consider themselves experts because they can use it, even though it's took them a couple of hours to get to grips with it.
No, that's really fantastic. It feels like Firefly is designed to be accessible to researchers of all experience levels, which I think is quite crucial for the adoption. So, we know that this is a constantly evolving field. So, Paul, how does Firefly support labs navigating a more dynamic environment?
Yeah, that's a really great question as well, because there's new techniques coming along all the time, new applications, new kits. There's a lot of vendors out there bringing great technologies to the market. So it's a really open platform, and it's really easy to set up new protocols. And we've got a big field application scientist team who actually go out and support our customers and help them develop new methods. And we work in collaboration with vendors like Scale. We also have a community through the software. So when we develop new methods, we post them on our community. All our users can download those methods straight to the Firefly. So yeah, it's we've tried to make it as open as possible to be future proofed as possible, and people will use it for many different applications. So the more applications that we can provide customers, and the easier it is for them to program themselves, then we just see it as a growing tool that's used for multiple applications in the lab.
Absolutely. Another thing I wanted to touch on is sustainability because it's becoming a bit of a major concern across research labs. So, my question, Paul, has to be how does the Firefly workflow address these concerns and look to promote kind of sustainable practices?
Yeah, that is another great question that's becoming increasingly important to try and reduce the amount of plastic that's being used. If we can reduce reagent volumes as well, that can help. I think one of the biggest things on the Firefly is that it has a non-contact dispenser. So an awful lot of applications that we develop are mostly additive steps. Once you've done a sample transfer, maybe adding barcodes and things that are individual well by well things. A lot of the reagents are then added to all wells. So the non-contact dispenser is able to address all the wells without any contamination issues from a single syringe. So you versus using lots of tips to do that. So the plastic saving is actually huge when you look at that, of using that dispenser versus pipettes. So that's one of the biggest single things that we've got. We are also looking at things like our tip packaging and so on to see where we can improve, reduce the amount of plastic, look at more recyclable materials and so on. So that's an ongoing project, but we're very focused and aware that we keep striving to do better.
Fantastic. So this technology seems to offer a lot of advantages. Giovanna, which specific applications or therapeutic areas do you see benefiting the most from this advancement?
I think with both of our platforms, um, we are developing them to be platforms to be used in many different areas of research and therapeutic areas. So it's hard to pick, just one, especially with an automation platform that can be used by lower throughput customers and higher throughput customers too. And that's really the types of products that at least I like to develop, is that really wide breadth of application areas. But maybe to pick on one, especially in single cell, those who are demanding higher sample numbers and higher cell numbers are really doing functional screenings or drug screenings, perturbation screens. And in these types of workflows, it's not necessarily the number of samples that they want to run, but they want to run many, many, many conditions over many cells. And when you're doing drug discovery, you need to ensure that you are minimizing your batch effects at all costs. And so to put these workflows onto an automation platform will help to reduce that batch effect and that user to user effect, on top of how we've designed our products to also reduce that down. And so, most of the customers that we are partnering together with are more on that drug discovery side of the world for their applications.
Wonderful. It seems like this has potential to revolutionize various fields of medicine as well. So, looking ahead, what does the future hold for this collaboration between researchers and companies like Scale? Um, let's start with Giovanna. Could you share your thoughts on what the future might look like?
Yeah, so if you don't mind, I'm going to go back to the past for a second because I really want to call out the team that has Firefly, and how collaborative they were and how easy they were to work with. We know that we specialize in single cell, and there's going to be other companies that specialize in other parts of the workflow. And, our philosophy is to, really work together with all of those collaborators, including the customers, but including the companies as well. We like to call it "Let's scale together" because we really do need to scale this industry together. And so I think just the partnership from the past was such a good foundation and example of how well this can look. So in the future, at least what I see is, we have a platform of products. Our first product that we put on the automation platform was our single cell RNA-seq workflow. We have our CRISPR workflow. We have a methylation workflow. We have new foundational technologies that are coming out that we'd like to expand on. So anyone who has a Firefly or any other automation platform that they have, can download those scripts from our full portfolio. So this, to me, just feels like scratching the surface of what we can do together, and we'll continue to put out more and more products together.
Fantastic. And Paul, what about yourself?
Yeah, much of the same, I think. Yeah, I don't think there's too much to add to what Giovanna's just said, other than I think what I've recognised is I think we're both companies that really listen to our customers, and I think we'll keep on doing that as well, and I think that will guide us. But yeah, I think we'd like to do. There's plenty more to do, and we'd like to get as many different applications onto the Firefly as possible. So yeah, bring it on, really. Yeah, happy to do more.
Fantastic. I think that is a lovely way to end this podcast. So, thank you both for sharing your valuable insights. This has been a fascinating discussion. And if you're interested in learning more about single cell research or the Firefly platform, be sure to check out the links in the description below. Thank you again for joining us today.
Thank you so
Thank
much.
you.